carbon molecule acetyl-CoA is funneled from the pyruvate. This oxaloacetate combines with this, and you get citrate, which is first molecule in the synthetic process. Then citrate is interconverted into isocitrate in presence of enzyme aconitase. And here, the actual trapping of the energy charge or trapping of the actual potential of the molecule starts. You have isocitrate dehydrogenase, which is forming NADH. And here, what you see is that one of the carbon molecule which is entering here is leaving here in the form of CO2. I have shown it in a different color. That means the same carbon which is entering is not the one which is leaving, but the point here is that one of the carbon dioxide is being leaving here. That means the oxidation process has occurred in this reaction. So what happens that NAD plus, which is oxidized, is reduced, and then you have one CO2 which is being left from this stage. Next is alpha ketoglutarate to succinyl CoA. Here again another NADH molecule is formed, one CO2 is being released. So these two steps you see are dehydrogenation and decarboxylation reaction, which we have already talked about. Then third step where NADH is being produced is a malate dehydrogenase. So you see there are three steps where NADH is being produced. One step is where you find that instead of NADH, one molecule of FADH is being produced. And also you find that there is one step here, you get one GTP molecule, which is equivalent to one ATP molecule. So what you have learned is that there are three stages where NADH is being produced, one place where FADH2 is being produced, and one molecule of GTP is being produced. Now this cycle will continue only if these NADH molecule is reverted back to NAD+. Otherwise, this cycle will not operate. And that's why it is important that the NADH should be oxidized. And that part we will discuss when we are going to talk about oxidative phosphorylation. So now the question comes, how this cycle is being controlled. So that is what you are going to learn. This cycle is being controlled mainly at these 2 points. These 2 points are the initial points where NADH is being produced. So first is isocitrate dehydrogenase, another is alpha ketoglutarate dehydrogenase. So these 2 points are the major point where the control occurs. And how it should be controlled? It should be controlled if there is a more ATP, this reaction should not happen. More ATP, this reaction should not happen. More amount of NADH in these two reactions, this should not happen. Less amount of ATP or high amount of ADP should activate this process. So let us see how these two reactions are being controlled. So they are the ones which are controlling the reaction rate of a TCA cycle. That means isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase complex. These two are important point where the major control occurs. Another point you will learn is at the point where acetyl-CoA enters in the here for the formation of citrate. Now, isocitrate dehydrogenase, this enzyme is controlled allosterically by ADP. ADP shows that the energy is less and more cycle should operate. And that is why what will happen that if ADP is there, they will allosterically bind with isocitrate dehydrogenase, activate this enzyme. And how it does, it changes or enhances affinity of enzyme. So affinity of enzyme increase means enzyme will bind more with isocitrate, NAD plus, magnesium, and ADP, and ultimately what you will get is you will get NADH. Now, if NADH is produced, what it does is that inhibits isocitrate dehydrogenase. And how it does? It does by directly displacing NAD plus. Now, ATP is also inhibitory. So what are inhibitory? ATP, the product of reaction, and NADH. It is important to note that several steps of the cycle requires NAD plus and FAD, and if they are in abundant and which are abundant only when energy charge is low. So if energy charge is low, that means your NAD plus or FAD is more and more cycle should operate. If it is less, or if NADH is more, then they should inhibit these reactions. Another enzyme which is being controlled is alpha-ketoglutarate dehydrogenase. This alpha-ketoglutarate dehydrogenase plays same role as pyruvate dehydrogenase. Both of them, they have homology. Here, alpha-ketoglutarate dehydrogenase is inhibited by succinyl-CoA and NADH. So what are these succinyl-CoA and NADH? You see that succinyl-CoA is the one which is synthesized at the later stage. So succinyl-CoA is the one which will be inhibiting this reaction, and same way the reaction is also inhibited by NADH, which is the product of the reaction. So it means that alpha-ketoglutarate dehydrogenase is also inhibited by high energy charge, and thus the rate of the cycle is reduced when the cells have high level of ATP. So actually what you are learning is that if ATP is more, the cycle will not operate. If ATP is less, cycle will be promoted. So this gives you overall mechanism. In some of the bacteria, the funneling of acetyl-CoA, converting pyruvate to acetyl-CoA is also inhibited by ATP, acetyl-CoA, and NADH. So what are the ATP? ATP is the product of the reaction. Acetyl-CoA is the product of reaction. NADH is the product of reaction. So all of them, they are in abundance. They will stop this reaction. Further, here also you see that ATP, which shows high energy charge, and NADH, which is a product of the reaction, they inhibit isocitrate dehydrogenase. And what activates is ADP, which acts as a substrate. Same way you see at this point, ATP is an inhibitor which shows high energy charge, succinyl-CoA, which is a product of the reaction, and NADH, which is also product of the reaction that inhibits. So actually what you are looking at is that the product of reaction is inhibiting the enzymatic activity. The synthesis of citrate from oxaloacetate and acetyl-CoA carbon unit is an important control mechanism in many bacteria. So this is also, so actually it starts even at the start of the TCA cycle before the acetyl-CoA enters. Still the control can happen in some of the bacteria. ATP is allosteric inhibitor. Allosteric inhibitor, you know that these enzymes, for example, citrate synthase is here. What it will do, citrate synthase is here. So citrate synthase, what it will do is that ATP acts as an allosteric inhibitor. It will not allow the synthesis of citrate. And the effect of ATP is to increase the value of Km. So if I increase the Km value, that means you require more substrate to bind, and that is the way the control happens. So it is very clear that at high level of ATP, the cycle will not operate. If the ATP level is low, the cycle will continue. Now, one more thing which is notable in the TCA cycle is that TCA cycle operates and if some of the intermediates are removed for the biosynthetic aspect, I will talk about that, and we have also discussed in previous class that some of the intermediates, they can also go for biosynthetic reactions. And if that happens, the TCA cycle operation will stop. What I mean to say here is that if I remove citrate in the form of fatty acid, if I remove alpha-ketoglutarate in the form of glutamate and other amino acid or purine, or succinyl-CoA in the form of porphyrin, so what will happen? Or aspartate, excuse me. So if I remove the intermediate in the form of amino acid, other amino acid, fatty acid, other intermediate, the cycle cannot replenish oxaloacetate. If it cannot replenish oxaloacetate, then the cycle will not operate. So under these conditions, what happens? The citric acid cycle operate only when the oxaloacetate is replenished. So cycle has to continue. If cycle has to continue, oxaloacetate has to be replenished. So in many cases, if suppose some of the intermediate has been removed, there is another way by which oxaloacetate can be produced. That is presence of pyruvate. Carbon dioxide is a fixed. That means you have this carbon, 3 carbon, 1 carbon is a fixed in presence of energy, and it can produce oxaloacetate. So this reaction happens in presence of ATP. That means it is an energy intensive process. That means it will happen only when the cycle operation is important. So oxaloacetate at catalytic amount will be produced, and the enzyme here is pyruvate carboxylase. So once pyruvate carboxylase is there, it will convert pyruvate into oxaloacetate. It's a carboxylation reaction in presence of energy. And That way, the cycle operation will happen. So this oxaloacetate, if I keep on producing, suppose I remove citrate in the form of fatty acid. Now I have to replenish it. So pyruvate, whatever the pyruvate is there, some part of pyruvate will synthesize oxaloacetate and the cycle will continue. Otherwise, if I block it, and even if I keep on feeding acetyl-CoA, the reaction will not happen. And last point which I want to discuss about here is that the cycle is operational, and on one hand it is producing energy, on the other hand it is also synthesizing important molecule. So this cycle is amphibolic cycle. That means it is involved both in catabolic process as well as anabolic process. So with this, I have been through with the TCA cycle. The next thing which you will be learning is about oxidative phosphorylation. So NADH or FADH2, which has been produced in the previous stage of the TCA cycle, has to be, utilized in the form of ATP. So actually, though we say that TCA cycle operates under oxidative condition, actually you have not seen any step where oxygen is being utilized. So how the oxygen plays a role? Oxygen plays a role by converting NADH and FADH2 into NAD plus and FAD, and this is the one which is required for continuation of citric acid cycle, and that's why citric acid cycle is considered to be an aerobic process. Under aerobic condition only citric acid cycle will operate. Why I'm saying so is you need terminal electron acceptor. If you can replace terminal electron acceptor, you can still produce the energy. But here, the most conventional electron acceptor is molecular oxygen. So today in this, we will also discuss how this process happens. So what you see here is that, let me just explain it, that so what you have learned is that we are going to talk about oxidative phosphorylation, where oxidation happens. So what is getting oxidized? Your NADH and FADH2 is converted into NAD plus and FADH2. So you have oxidation process. Here, electron transfer you will learn. I will talk about how electron transfer happens. And during this process, one proton is being pumped out. So once proton is pumped out, there is a difference in the charge across the mitochondrial membrane, and this proton re-enters, and the re-entry doesn't happen from anywhere in the mitochondria. It happens only through enzyme which is known as ATPase enzyme, and this enzyme allows proton to enter, and the energy which is responsible for this membrane potential is the one which will be responsible for synthesizing ATP. Though there are many more processes, many more hypothesis which has been given for ATP synthesis. One of very conventional is that the potential between the outer mitochondria and inner mitochondria, when proton enters, this difference in the electrochemical potential is the one which is responsible for synthesizing ATP from ADP and PI. Another very conventional process is that it is not the electrochemical gradient which is responsible. Rather, when proton enters, the structural changes occurs in the ATPase enzyme, and that is what leads ADP and PI to come close to each other, and that's the way ATP is being synthesized. So there are two major hypotheses by which ATP synthesis has been shown to be coupled with electron transport process. Now, your question may be that if I uncouple it, suppose I allow all the proton to leak out of the mitochondria, will this process happen? Answer is no. If we decouple mitochondrial electron transport with the phosphorylation process, this will not happen. Suppose I block the synthesis of ATP here, that will not happen. If I stop the proton entry inside the mitochondria, the oxidative phosphorylation will not happen. So there are many ways in which this process, oxidative phosphorylation, does not happen, or ATP synthesis does not happen. Suppose I block this reaction where oxygen is acting as a terminal acceptor of electron, still the process will not happen. Oxidative phosphorylation will not happen. Your ATP will not be generated. So if I look at this reaction, it is again a depiction. It is happening in the mitochondria. You can see in the left side, the mitochondria, which is present and which is shown in the fibroblast cell. So this shows you how the mitochondria looks like. You have inner membrane, outer membrane, and what you have is that citric acid cycle operates. Now the citric acid cycle will produce NADH. This NADH will go in electron transport. So you have different, proteins which are present on the mitochondrial membrane. On the inner side, they will be, transferring the electron, and then there will be proton which will be pumped out of the mitochondria. These protons, they will re-enter in the mitochondria and they will synthesize ATP. So a simple process. Citric acid cycle is responsible for your electron transport because it is generating NADH. And why NADH? Because NADH is the one which has high electron donating potential. Then mitochondria, it is general, physiology, where mitochondria has outer mitochondrial membrane, it has inner mitochondrial membrane. Inner mitochondrial membrane is highly folded, and it is the one. This is just the depiction of how mitochondria looks like. It has a very high surface area where you have cristae and two compartments in the mitochondria. One is the intermembrane space. So you have outer mitochondrial membrane, inner membrane, and then you have a space between outer and inner mitochondria. The matrix, which is bounded by inner membrane. So inner membrane is having inside, you have a matrix. Oxidative phosphorylation takes place in inner mitochondrial membrane. So this is important. Oxidative phosphorylation takes in the inner mitochondrial membrane. In contrast to most of the reaction of the TCA cycle and fatty acid oxidation, which it takes in matrix. Though it is not far away. So matrix is here, the TCA cycle will happen here, and the oxidative phosphorylation will occur in the membranes of the mitochondria, inner membrane of the mitochondrial system. So here, of course, I'm not going to talk about porine, etc., which is present. It is pore-forming protein which is present in the mitochondrial membrane, which is responsible for flux of phosphate, chloride, and organic anion, as well as adenine nucleotide. I'm not discussing about how ATP and ADP is controlled, but it is worth to understand that most of the ATP utilization process is outside the mitochondria. That means most of the synthetic process, they will be happening in cytoplasm. And for example, protein synthesis is happening on the surface of ribosome, which is in the cytoplasm. Many other processes, biosynthetic processes, will be happening outside the mitochondria, but energy is being produced inside the mitochondria. This oxidation due to phosphorylation will, as I said, will depend on the electron transfer. So you have NADH, FADH2, which is responsible for, which are reduced, and they will go to the molecular oxygen. So actually, molecular oxygen is reduced to water molecule. So that's the way electron acceptor works. And high energy electron is acting as a redox potential. So we will also talk about your electrochemistry, which you have already learned, that how electrochemical principles are applied over here. So high energy electrons, they are the one which are coming from NADH and FADH2. And in oxidative phosphorylation, electron transfer potential of NADH and FADH2 is converted into phosphoryl group transfer potential. So actually, it is the energy transfer, your potential of electron transfer, which is present in NADH, is going to be transferred to ATP molecule, which becomes ATP. And what is ATP? ATP is the one which has a high phosphoryl group transfer potential. The measure of phosphoryl transfer potential, is given by delta G0 prime.
Excuse me, sir. So we have another quiz at 9:50. So, we have half an
How
hour.
come you can have a quiz at the time when you have, exam? This is a class time. You cannot have another quiz. It is
So we are
impossible.
asked to log into Moodle at 9:50 and
Mhm.
put the password for the quiz. So we'll have to end the quiz by 9:50 or this quiz.
This quiz will be ending. I will be starting at 9:30. You will be ending at 10 o'clock. 20 minutes quiz.
So we have another quiz at 10 o'clock itself. Like on 9:50, we have to log in into Moodle and enter the password. It would be practically
You see, I
impossible,
cannot
to.
help it. I have announced it. I will continue it at this time only. Okay. This is the class
So
time. You
that
cannot
quiz
tell.
was
You can tell
scheduled
your
like
teacher that.
5 to 10 days before.
Yeah.
So that quiz, which is on 10 o'clock at Moodle, was scheduled 5 to 6 days before. We can't tell them to, postpone it because
Look,
we have
then you do at 10
0 minutes.
o'clock. I'm not saying quiz is a quiz. It is in the class time. I'm not taking any extra time. So 9:30, I will start, and 10 o'clock you have to end. You can end it earlier also. I'm taking it in class time. I'm not taking any other's time. So anyway, I'm stopping this. It is very disturbing that most of you don't like to be present in the quiz time. I'm just going to upload, and you have to, by 9:30 you can start, and 10 o'clock you can end it. So I'm sending this for quickness to your, email ID, and you can upload on Microsoft Teams. So I've sent it to your, class ID, and I'm also uploading this on your Teams. You can submit in Teams. Have you received it?
Yes, sir.
Okay.
Sir, can we leave the meeting?
You can leave the meeting. You can start your quiz. And then you can submit it in Teams only.